Well, thanks a lot, Michael, for this very kind introduction. Also, thank you for the invitation to come here, for me the first time in Poland, in Krakow. Such a really beautiful city. You're really to be envied, for such a lively environment here. So my global theme today is cell biology at work in the awake mouse brain. And I picked this as a sort of an envelope theme for this lecture because if we think about from which sort of evidence do we derive sort of all our ideas of how the brain works, we have to conclude that actually a majority of these data comes from very reduced model systems. And the question is, to what extent can such model systems deliver, answers to the problem of how the brain works? And I want to illustrate that with the following cartoon. So this is like a cultured hippocampal neuron, which has been placed in the desert. It's a really deplorable, poor, structure sitting there, completely deprived of all its environment that it usually has in the brain, with all the glial specializations, all the other fellow neurons, and so on and so forth. Lots of things that neurons actually, I guess, need to proper function are not there in such kind of very reduced systems. So the question really is, what do we learn from studying these kind of systems for how the brain in the end works? So I think that one thing one needs to really do systematically is to at least study fundamental mechanisms in vivo and then try to build bridges to the reduced systems so that at least one can see, well, this feature of whatever synaptic plasticity or organelle transport or whatever does work like this in vivo. It also does work like this in the reduced system. And so maybe we can then use the more reduced systems that, of course, are more easy to do manipulations and so on to study such things. So, and obviously, there will be also many questions that can only be addressed properly in vivo, and I'll show you also examples for that. So I wanted to illustrate this case for 3 examples that are listed here. So one would be organelle trafficking. And the second will be the quantitative measurements of ions in vivo, and then a third example will be central structural plasticity, so how neurons change their structures in response to certain stimuli. So those are the 3 topics I want to cover, and I think Michael also asked me to explain the basic methodology a little bit. I guess most of you know how one typically addresses these kind of questions. One is you have to, of course, get access to the brain, so you can do a craniectomy, for example, as shown here. So you open up the skull, then insert like this window here, cranial window, attach this crown, for example, as a mechanism to clamp the head firmly to this kind of stage here. And you can see this mouse has been habituated to be in this situation, and you can see this kind of disc here. So this is a turning disc. This is one of many strategies how you can make the mouse feel as though it's kind of free, it can run, and that's a very important thing so that it feels happy. And so you can now basically look into the brain in the awake animal. And how we do that, this is using 2-photon microscopy. I'm sure that most of you know about this, but I'll nevertheless just point out some of the key features here. So this here shows you single photon excitation with blue light as shown here. So you excite your fluorophore, emit green fluorescence, and you can see that this fluorescence is distributed in this hourglass-like shape. So you have a huge volume where you excite molecules, meaning that typically people think you get the signal from the focal plane here, but it's actually only 50% in this kind of wide field imaging approach that comes from the focal plane, the rest comes from elsewhere. So you can, of course, block this off in confocal microscopy by limiting the light to this spot, but I think the even better way to do this is 2-photon excitation, which is shown here, using infrared light that you can see again here. And if you use, let's say, in this case, 800 nanometer light, you can, of course, produce a very high intensity of this light in a very small volume. This is sort of the illumination volume here. Then you get a high enough sort of density of photons so that this highly nonlinear process of two-photon excitation happens, and then you emit a green light and can image that. And this is the reason that you get only emission here. There's a magnified version from this very tiny spot. Now, obviously, you can scan this spot in your sample and thereby generate an image, and it's formidable for optical sectioning. And sort of an advantage that comes with it is also that you use infrared light, which is very penetrable, so it goes very deep into the tissue without being scattered too much, since that depends on the wavelength of the light. The downside is that the resolution is a little bit less than if you use, like, let's say, blue light for imaging. All right, so this is sort of the basics, just for those of you who are not so familiar with this, of this microscopy technique. And I'm going to show you here a small movie where we now start from the surface of the brain and we just focus in down to 500 micrometers into the brain. And you will see two structures here. So you will see in red thalamocortical boutons that we have labeled. So they come from the thalamus and form synapses in the cortical layers. And then you see here in green a single pyramidal neuron that we stain with membrane-bound GFP. And so let's let this run here. So this is now layer 1. I guess now slowly layer 2 is approaching. So you can see this dendritic tree here. You can see this really neat optical sectioning property of this technique. Here, this is the soma with this membrane-bound GFP. And then as we get deeper, towards 500 or below, the signal is getting less strong. So this is how it typically looks like, this kind of data, so that you have an idea what we're talking about here. All right, so let's start with the first topic here. This would be dense core vesicle trafficking in vivo. So why is that important? I mean, dense core vesicles is maybe a bit of a neglected field, not that many people working on that, but it's actually a physiologically extremely important organelle because it mediates a whole bunch of physiological functions. For example, of course, neuropeptides are released, neurotrophins are released and transported by these electron-dense vesicles. You can see this is basically the membrane and the electron-dense core. Guidance cues are transported, proteolytic enzymes, components of the extracellular matrix, transsynaptic signals, maybe even active zone precursors, although that idea I think, is a bit more under siege these days. So a lot of functionality that is mediated by these systems, and it seems that there's also pathology involved in kind of misfunction of dense core vesicles. And so one is social anxiety disorder that seems to be related to oxytocin as a presyn transport and release via such structures. Stress disorder seems to be linked to NPY, and neurodegenerative diseases might also be related to malfunctions in this regard. So it's a very widespread, important system throughout the brain that is implemented by these dense core vesicles. Now, dense core vesicles are typically produced in the soma of the cell here, and then transported out to the axon and along boutons, so these labs here are boutons that the axon forms, and then all the way to the tip, and then things kind of return back and come back to the soma. This is sort of the typical idea how one thinks of dense core vesicle trafficking. And one thing that is different from synaptic vesicles in this case is that dense core vesicles are only synthesized here, and there's no recycling going on in the periphery, so they always have to be redone from scratch. So that's why you need the soma and all these intricate transport processes in order to deliver this cargo that is transported by these vesicles to their sites of action. And this is actually also something that is not well known in this field. So are there dense core vesicles that only have NPY or only have whatever BDNF or any other of these things? Or is everything kind of mixed up in all of the dense core vesicles? So this is very unclear. Could be populations of dense core vesicles, could also be that they're all just containing whatever the cell produces in terms of potential cargo. Now, one thing in terms of their release is that they're preferentially released near synapses and also extrasynaptically, but this doesn't happen so frequently. And also the stimuli that are required to release dense core vesicles are actually very unknown. In cell cultures, one needs something like 100 Hertz stimulation for a little while in order to produce a strong enough signal so that dense core vesicles really release their cargo. But in vivo, this is totally unknown. We don't know what the stimulus configuration is that leads to dense core vesicle release. I mean, there's one, just a little sideline. When people use optogenetics and stimulate neurons at 40 Hertz, often frequencies for a little while, and then they see all sorts of effects. Could be dense core vesicle release. I think people don't really think about this too much because they think it's normal synaptic transmission that they stimulate with this. But maybe these paradigms specifically release dense core vesicles. But whatever. So trafficking of dense core vesicles has only been studied in culture neurons, at least for like the mammalian system. There's one in vivo study that has been done in Drosophila. So this is our starting point here. So we want to image dense core vesicle trafficking in the living mouse brain in anaesthetised and awake states. And we want to determine the typical parameters for dense core vesicle trafficking, like their speed of transport, their directionality of transport, pausing, and so on and so forth. And of course, we also want to identify the native conditions for dense core vesicle release. I put this in gray because we haven't managed to really get any reproducible results on that end yet. So the approach to address these goals is in vivo fluorescence labeling of dense core vesicles using cargo GFP fusions, implantation of chronic window, in vivo two-photon microscopy, and then semi-automated tracking analysis of the dense core vesicles and their characteristics. We also did some experiments using generorhodopsin stimulation of fluorine-intact dense core vesicles that one could use to identify release events in the intact brain. We'll see one example later. And then, of course, in the long run, we want to do physiological simulations to see whether, whether those could release dense core vesicles in a predictable manner. So this is our labeling approach here. So this is like this typical EM picture of a dense core vesicle, which contains in the center chromogranins A, B, and C. These are like specific proteins for dense core vesicles, and they contain cargo, as I mentioned before, neurotrophins, neuropeptides, and so on and so forth. And we now use this NPY venous fusion protein. So this little thing here is NPY, and here our beloved GFP fusion protein. And then we can imagine that a number of molecules will be packaged into every dense core vesicle. This is just arbitrary. I have no idea how many GFP copies would be inside such a vesicle, but it's obviously enough to image them directly. So we use a viral expression strategy to get these expressed in the brain, and use this AV12 virus that we've been using for a long time, and use here synapsin-driven NPY venous fusion constructs. And at the same time, we express M-cherry. And this allows us to label these thalamic projection neurons, and we chose these because the injection site is nicely remote from the site of imaging, and because these axons typically form in the superficial layers, in the molecular layer. They form like a parallel sort of branching, like illustrated here. And this, of course, for trafficking analyses, you can image basically in one plane, and you can see how the vesicles go along in that one plane. So that makes imaging very easy. And by having both vesicles and the axon labeled, we can, of course, single out individual axons and then follow the vesicles inside that axon. So there is a certain imaging challenge here that I want to illustrate with this cartoon. So this is our objective. Then we're having those superficial labeled axons with their dense core vesicles. This is the size of a dense core vesicle, roughly 100 nanometers. This is the focal volume or point spread function of our illumination. So about 1 micron in Z and about 350 wide. So this is about the volume that we excite and from which we collect fluorescence. And now basically if you put 3 such vesicles in the illumination volume, you would get a certain intensity of a spot that you would see in your imaging, in your image. And then if you, let's say, do it like this, you still have 3 vesicles, but they're differently oriented. The outcome will be the same. So what you see with the microscope, same things. You can't discriminate that. And then if you have only 1 vesicle sitting in there, then most likely this gets a little bit dimmer. So if you kind of go back and forth, you can notice this difference in the ideal case. Yeah. So this might be one way how one could potentially discriminate how many vesicles are in that volume, but we tried to do some sort of quantal analysis. It didn't work, so there are too many factors that influence the intensities of those signals. So now if you imagine there is an axon running through your focal volume that you're imaging, and you have in blue, this would be autofluorescence, this would be your specific fluorescence. Now, in this situation, you could easily discriminate, obviously, this vesicle from that, and it would give rise to a signal like this, for example. Now, if you have this situation, let's say multiple axons running through that focal volume, then, of course, the signal would be maybe a bit brighter, but you cannot discriminate, that these are 2 axons. Yeah? So this would be the problem that we face with this imaging approach. And knowing this, we deliberately generated a situation where we have only very few labeled axons, so that there's a lot of distance between individual axons, so that we don't run into this problem, but that we can assume that this is our imaging situation. So this is now showing a typical Z scan that we obtained here with these axon profiles that one can see here. And this is the PL surface of the brain, and this is the depth at which we typically do the imaging. And so if you now take an image plane along this line, this is what you will see. And if you zoom in a bit, like done here, you can see, for example, here 2 axons that contain dense core vesicles, and now we can start imaging them. So this is an example that is sort of probably the best one that we could obtain in all these imaging sessions. What you see here is an axon that runs like this, and you see this synaptic bouton here, presynaptic bouton. Then there's other axons also here, and then there are some green vesicles where we didn't get the axon stained. That can of course happen, that you don't get 100% co-expression of the 2 things. So that's why there is also some green vesicles. So now let's look at this. So we did a time lapse where we recorded vesicles in every once in a second, so at 1 hertz, and you can see how these vesicles, go in both directions through that axon. There's another one here. You can see that they sometimes pause, and then they keep going. You can see there are sort of traffic jam areas, like one could see it here, where vesicles from both sides come and then briefly stay, and then they continue. So that's what's going on in your brain right now in, zillion-fold times, exactly like this. So now let's look at a different example. So this is at the other end of the spectrum. So this is an example that is not, in terms of optical imaging, not ideal, but still analyzable. And you can see there's more background fluorescence lingering around here, all these green kind of dots that come and go. But you can still see, we can clearly resolve trafficking vesicles here. You can also see that here the motion correction didn't work as well as in the previous example. However, I think the basic principles are still the same. You can see those presynaptic terminals here, and we can see vesicle trafficking going on in different directions. So this is the data that we obtained and that we now analyzed by doing these typical chymographs. This is a so-called chymograph down here. So what we show here is this is a stretch of axon that we imaged. So the transport in this case goes from right to left. There are 3 vesicles at time point 0 that are circled here with different colors. And now if you look at this one here, you can see that for 2 seconds, 4, and so on, as time progresses, it moves a little bit, a little distance. So distance is basically in x and time is here in y. And so you can then see, according to this trace here, that shows you the trafficking kinetics of that vesicle. So you can, in the end, analyze these kind of lines here in order to get quantitative measures. The more vertical or the more steep the slope of these lines is, the slower they move, the less distance they cover in a certain amount of time. For example, this blue vesicle here seems to fuse with the yellow one, and then they seem to migrate both at the same speed. And then this is illustrated by this line. So this is a more flat line, meaning the vesicles cover more distance in a shorter amount of time. And so we then analyzed lots and lots of those tracks. So typically we recorded like 10 minutes for each of these, and then we analyzed in total way over 60 to 80 of these of such tracks. So then you can, of course, obtain mean speeds, about 1 micron per second movement. This is like average movement. So these are these whisker plots. I'm not sure how familiar you are with these. So this is basically the 25 to 75% interval, and this is the 2.5 to 97.5% interval. And these dots are outliers, so that just that you see, can judge the distribution of the data. So then we looked at the max speed that they can attain. So this is like 2 microns, but there are a few vesicles that even make it up to 5 microns a second. Then we can look at the fraction of pausing time. So how often do they stop and remain during such a track here? For example, this might be like something like a pausing time here. Then it moves and then it pauses again. So this is about 10%. And directionality is simply a factor that expresses whether there is a preferential movement in one direction. For example, a factor of 1 would indicate that all the vesicles go into one direction. And a factor of 0 would then indicate that they go in the opposite direction. And 0.5 would be that that both directions are about the same likelihood. So now we can compare these data with data that has been obtained in cultures, and you can see that the match is actually pretty good. So it's 0.78 was here recorded at 32, 1.14 at unspecified temperature, and 1.27 at 37. So these values match very well with what we have found in vivo. Here, this pausing time in vitro seems to be more pronounced as it is in vivo. So this could be now one first difference that we notice here. So the next thing that we looked at was microtubule dynamics. So you can also watch basically how microtubules are synthesized in vivo. And the way we do this is to use MAC-F18 GFP fusion protein. This is like a minimal fragment of the microtubule actin cross-linking factor 2 that gets selectively incorporated at the plus end of the microtubuli. So by then watching where the incorporation of this GFP-labeled MAC-F happens, we can actually derive the transport direction because the plus end is in the periphery, minus end of the microtubules at the soma. So this shows again like a stretch of axon with this MAC-F signal. Not very illuminating in this state, but now let's see and focus maybe on this red thing here. So this is basically where MAC-F gets incorporated. Then 44 seconds later, it's here. Then it gets incorporated here, then there. And this is actually referred to as comets. And I think from this, it's a bit difficult to see why this is called comet. But if you imagine the trace that this action leaves behind, this is basically a microtubule that is now, let's say, has started here. And now it's being built into this direction. So you can see the microtubule grows. But of course, you only see the action where the plus end extension is taking place. So basically, this is showing us the direction of microtubule production in this axon. So from this data here, we can now say, well, this is the minus end and this is the plus end of that axon stretch. And of course, you can do like these kind of chymographs again. And you can see the lines are pretty steep, meaning that's very slow transport here. Some of them are just residing at one situation as shown here. So this, of course, you can now quantitatively analyze, and we see that microtubule plus end extension is slower, so it's about 0.1 micron per second, so 10-fold slower than dense vesicle transport. The run length is on average something like 10 micrometers, so the run length would mean basically how long you can see this process in one situation. And the number of such comets per axon stretch is about 0.4. So, but now we can compare that, of course. It's very similar to in vitro, and we find actually fewer comets in the axon compared to dendrites, where that, of course, also happens. Now we can do both at the same time. So we can do imaging of microtubule plus end extension, and we can do dense vesicle imaging, and then basically that would allow us to identify the direction of transport for the dense vesicle, so we can discriminate antero versus retrograde transport. And here in green, this is basically this chymograph interpreted in simple terms, and you can see that all the green lines go from left to right, meaning that here's the minus end, here's the plus end. So basically this is retrograde, anterograde transport, and this direction is retrograde transport. Now if you look at the traces of the dense vesicles, you can see, well, this one goes anterograde, this one goes retrograde, and so on. So we can now identify or analyze selectively the anterograde transport speeds versus the retrograde transport speeds, and this is shown in these graphs. So we get here faster mean anterograde transport than retrograde transport, which probably could be due to different motors involved in this, and the max speeds also differ, and the pausing fractions as well. And this just shows basically direct comparison in the same axon, antero versus retro, and it's very clear that in all cases, retro is slower than anterograde transport. So now then we ask, well, is anesthesia in some way maybe important for defining or influencing transport speeds? Because everything I showed you so far was in anaesthetised mice, and so we then started to do imaging in awake mice, and this is how it looks like. So you can see it's more of a challenge to do these experiments in awake animals. Despite motion correction algorithms, there is still a lot of motion involved here. But still, I mean, you can see clearly vesicles coming along here. You can also track them. And of course, you have to use more strict criteria as to which stretches of axon, here one can also see it quite nicely, you analyze. And so we did this analysis, and we found that in the awake state, there was essentially no difference in the basic characteristics of transport, yet there was one thing that differed. This was the directionality. It seems that in awake mice, there's no such strict directionality going on as it is in the anesthetized. So how to interpret this mechanistically, I don't know. But that's an observation. All right, so then one thing that we also looked at, because we noticed that when vesicles actually approached these boutons that you can see here, that they seem to be slowing down, and so this is something one can see here. So there are kind of vesicles coming along here, and then they enter the bouton and slow down upon entry, upon sort of approaching the bouton, and this was quantitatively analyzed here. So these are, let's say, 3 boutons in this axon stretch, and then we have trace vesicles. So the red one comes in here and then stays in that bouton, and then leaves the bouton again, which can be also seen by this chymograph trace. And so we could then identify speed separately, externally to boutons and internally to boutons, and Not too surprising, the speed was slower inside the boutons. Max speed was also different. And the pausing fraction, of course, was higher inside the bouton because you would have to surmise that in the bouton, which is the preferred site of release of dense co-vesicle content, you have to have some mechanisms that interact in order to dock these vesicles and make them fuse and release their content. So yeah, this is then this graph that shows you then the vesicle approaching the bouton, and it seems that already outside the bouton they start slowing down before they then enter the bouton itself, which can be seen by this curve here. So that is also an observation that hasn't been made before. So now this is our challenge at the moment to determine the physiological paradigms or parameters for dense co-vesicle release. So we have here NPY fluorine. So fluorine is a pH-sensitive GFP variant that would basically blink up, so it would get brighter as the vesicle opens and the pH gets equilibrated, and then the content is expelled into the extracellular space, and then the signal will disappear again. And this is what we saw in a very few cases after stimulating with general rhodopsin that we expressed in the same axon. And if you focus on this area here, you will, so this is the imaging situation, and here at this side you will see it after this black frame, which was a blocking frame because we applied the light for optogenic stimulation. There was a small signal, and this is magnified here. So here you can see this is going up, and now suddenly it disappears. So this is most likely a dense co-vesicle fusion event. But that is the only one we found so far. It's very hard to detect them and specifically find them. So, of course, one could now examine this, for example, in acute pain, and because it's known that certain peptides play a role there, we could look if that, for example, could trigger specifically the release of these vesicles. So this is the first summary here. So we established an approach for in vivo imaging of dense co-vesicles in mice. We show for the first time in vivo dense co-vesicle trafficking characteristics in mice, and also microtubule dynamics in cortical axons. And we show that the movement dynamics in general are similar to in vitro study, but that there are differences in terms of the pausing fraction and in terms of the interaction with boutons. And we could also show that the anesthesia does not affect this basic property of vesicle, dense co-vesicle transport. And we could confirm that the anterograde is faster than retrograde trafficking. This is still something we need to do. So now let's go to ion imaging. This is another quite challenging approach to measure concentrations of ions in vivo. And so here we are addressing the question, what is the resting chloride in neurons in vivo, which has not really been addressed to such an extent. Does chloride form intracellular somato-dendritic gradients in vivo? And is KCC2 required to define those chloride gradients? And this is, there's some debate going on in this field whether or not this is the case. I just want to illustrate that briefly again, although I think Eve has given you a pretty good introduction of chloride handling and the role of KCC2 in this. But the key point is that the polarity of GABAergic responses depends on the chloride concentration. So this is not a a priori defined feature that a GABAergic synapse does hyperpolarizing inhibition. So it can be hyperpolarizing inhibition, for example, if chloride is low, like depicted here. But these responses can even be depolarizing if the chloride is high, for example. They can also be in an intermediate range, let's say around 10 millimolar. You would get basically a situation where a GABA channel opens, but there's no chloride flux. So only then, let's say, if a glutamatergic synapse nearby would open or signal, then you would suddenly get something like that's called shunting inhibition. So this is very important to be aware that relatively small chloride concentration changes in soma dendrites of a neuron can fundamentally affect the polarity of GABAergic responses. And that's why it's important to look for these things. And so one hypothesis is that there are such gradients in cells, that there is low concentration here, so GABA would be inhibitory here, but that there's a higher concentration out there, which would make GABA depolarizing. And I think this is a particular challenge to examine in reduced model systems, because chloride is so strongly dependent on all sorts of variables such as potassium, pH, bicarbonate handling, and all these things, where I would say this you can only perfectly reproduce in vivo, because only there you have perfect control over all these properties. And basically all the factors available that could potentially influence the system. So how to do this, how to go about this, Eve already introduced the chlomelion yesterday. So there's a variant which is called superchlomelion that George Augustin has further developed, and we described this indicator already in 2000. I think this is one of the sort of first generation genetically encoded indicators that have been developed. And it's a protein that consists of cerulean and topaz linked with a linker. And the chloride sensing principle is based here inside the topaz, or the YFP molecule, which is inherently chloride sensitive. And this chloride binding, or the chloride binding effect goes, or exerts its effects on the fluorophore via protonation of the fluorophore. So basically, proton and chloride sensitivity are a closely linked issue in this sensor. Now, it's a ratiometric sensor, which means that you can calibrate the ratio that is shown here as a function of chloride concentration. Here, this is now cultured hippocampal neurons, where we can clamp intracellular chloride in a reasonable manner. And so we can add different chloride concentrations outside and add some ionophores that would then equilibrate inside and outside of the cells. And then you get these kind of calibration curves. So in the end, you can read out from a measured ratio in the cell, you can read out the chloride concentration. So this is how this works. And I already alluded to this sensitivity of the construct to protons, because the effect of chloride binding is mediated via the protonation, changes in protonation of the fluorophore. And the effect of unknown pH changes, let's say you have calibrated your indicator for pH 7 and you read out a certain ratio that you have calibrated with, to 5 millimolar chloride, as shown here. And now if your cell suddenly changes pH from, let's say, 7 to 6.6, then of course you will read out 10 millimolar chloride, although the chloride didn't change. So it was only a pH change. So this is something one has to be aware of with this system. This makes imaging a bit challenging with this indicator that it's both chloride and pH sensitive. So we're expressing this also with AAV viruses. So our approach typically is that we use this diocassette to express the superchlomelion. And since we want to have sparse expression, so not really tight expression, dense expression in the brain, just in the ideal case, a few neurons, or maybe one neuron per volume and nothing else surrounding it, because that improves the imaging conditions. We use this kind of dilution approach that we developed in the lab. So we co-inject with this construct here a Cree virus that we can strongly dilute, and the dilution basically determines then how many cells you will get express that indicator. So by using this cassette, you can use a strong promoter here that really gives rise to maximum expression levels. And as you know, in this expression system, the expression levels are strongly dependent on the multiplicity of infections, so that the AAV virus, that several virus capsids infect the same cell, and that really boosts the expression a lot. And now by using this dilution approach, you can take advantage of maximal expression here. But for the Cree, of course, you need a few molecules in order to exert the action. And so that's how you can easily control the density of expressed neurons. Now, this is an example that shows the topaz channel here of a neuron in layer 2-3 of the motor cortex. This is the cerulean signal here, and this is the ratio signal. And these small ratios would refer to high chloride, and the large ratios to low chloride concentrations. And so in some cases, I to emphasize that, only in some cases we had strong enough expression so that we could even resolve spines. This was typically not possible. And also only in a few examples we had this kind of imaging of basal dendrites, but you can see in the ratio image there's not much left to analyze here. So typically what we're referring our analysis to is this proximal dendrite and the soma of the neuron. I think these are areas that we can reliably quantify. And what we found is that there was a mean chloride of 6 plus minus 2 millimolar here in this proximal somato-dendritic compartment. And now if we look at the distribution of ratios that we imaged here, we were really disturbed by the observation that the ratios differed dramatically. So from 4, which would indicate a very high chloride, to 10, which would indicate a very low chloride. And this was very disturbing. On the other side, you can see that within a cell, if you compare basically the cell, different compartments of the cell, so here soma and dendrite, there was barely any difference. So they're basically the same. So what is that? So why is there such a big variability in readouts? Now this is just showing basically the distribution of these ratios that we read out, and you can see it's just like a Gaussian distribution that covers a huge range of ratios. So, of course, I alluded to this already before. This is most likely pH that plays a role here, and so we now use the superecliptic fluorine as which you can actually also use as a ratiometric pH indicator by exciting it with 2 photons, and then it emits actually at these 2 wavelengths, and you can ratio these 2 wavelengths and calibrate it in order to read out pH in cells in vivo. And we use the same expression paradigm in the motor cortex as explained before, and this is how it looks like. So here we have like the yellow emission and the bluish emission of the signal. You can see the signal to noise is not great. It's just about okay to work with it. And then we can calculate the ratio here, and from these ratios we can infer the pH. Now this graph shows the distribution of pHs at stomata of neurons, and you can see it's really wide. It goes from 6.5 to 7.5. So it's a huge range of pHs that is covered by different neurons. And now we think that this is actually also the reason for this big distribution, dispersion of chloride readouts. And if you compare those two, sort of this large range here is probably due to systematic shifts and changes in resting pH in cells. So this is something I would have never expected, and I was very resistant initially to believe this, but I'm quite convinced that this is actually really the case, that neurons obviously seem to be, present in vivo at really vastly different pHs. Whether that is related to different activity levels or whatever, we don't know, but I think that would be something interesting to analyze further. So that also in the end shows that if you want to really say for one single neuron it has this chloride, the indicator is good enough to deliver this, but as long as you don't know the exact pH in that cell, you will have a problem. Yeah, so basically, if one wanted to really accurately measure chloride in individual cells, one would have to do simultaneous pH recordings, and that's something we have not yet achieved. There's another indicator that people have described, which is named Clofensor, that can do both things at the same time, but that is very complicated to use. Okay, so also here we look briefly for effects of anesthesia on chloride, and you can see this is an anaesthetised. This is a wake state, and we again looked between soma and dendrite, didn't find any difference here, and as you can see here, there's also no difference between anesthetised and awake animals in terms of chloride internal concentrations, which I think is also quite surprising that there is actually no difference here. And then we have looked at the pH, whether there is differences in pH in anesthetized versus awake. And again, you can see the distribution is more or less the same. There's no difference between pH regulation in awake versus anesthetized. So then we wanted to look at KCC2 function in these cells, and what we've done here is we use the floxed KCC2 line, so like a conditional knockout line, and we then use the construct that I showed you before already, so the chlomelian construct, and we use this diluted Cree expression approach. So this results in a quite neat experimental situation. So we can inject on this side of the hemisphere, on the left hemisphere, this construct and Cree, and then the cells that would express the indicator would at the same time be knockout cells. So we have individual number of defined knockout cells that we can analyze here, and on the contralateral hemisphere we can just inject this one here, chlomelian without the dio construct, and then that would give us basically a direct comparison in the same specimen. And so we did that, and you can see here this is the result. If you don't express Cree, this is with Cree expression. You can already see that the topaz emission is much less intense than here. And if we now plot this here, first in the anesthetized situation, here we have large ratio, meaning small chloride concentrations in the presence of KCC2, which then very clearly shifts down to ratios around 5. That would suggest high chloride in these cells. And we do see the same shift also in awake animals. Now, the question would be, well, how does it look with pH? Are there changes in pH if you knock out KCC2? That's obviously not the case in both anesthetized and awake situations. So these experiments show that we can really now in vivo demonstrate that KCC2 is required to get the low chloride concentration in neurons. All right, so this briefly summarized that. So we have in vivo somatic chloride of 6 millimolar, meaning that GABAergic synapses will produce hyperpolarizing inhibition in this situation. We have in vivo somatic pHs on average of 7.1, but very large inter-individual variability, where one has to see what this is all about. In vivo chloride and pH are not affected by isoflurane anesthesia. Chloride is uniformly distributed, at least in the proximal somato-dendritic domain. We cannot really address this for the distal parts of the dendritic tree. So in a way that question still remains open to some extent. But in this, throughout this compartment, GABAergic synapses will be hyperpolarizing. And we also show that, low chloride, or in order to achieve low chloride, you absolutely need KCC2 expression also in the adult animal. And obviously, this is our sort of quantitative caveat here that we needed to develop now a strategy that would allow us to read out pH simultaneously with chloride in order to use at least this sensor for addressing such questions. And I think with the KCC2 experiment, we also demonstrated that one can actually one can detect differences in chloride concentration using this indicator. Of course, what you would ideally want to do is to really look at, functional readouts. So image inhibition as it happens along the dendritic tree, that would be the dream experiment. But I think that's still some way down the road to get there. So finally, I want to just have a few slides on a different topic, which is structural plasticity. So now we're asking, are there any structural changes in the cingulate cortex in chronic neuropathic pain? So you're looking here at an area which is the cingulate cortex, so ACC, anterior mid-cingulate cortex. The nomenclature, I think this has been pointed out by a previous lecturer here, is a bit complicated in this cingulate cortex. But what you can see here is that this region is really like one of the central regions, so many arrows going to this region for producing pain states. And pain, as you know, is a condition that is chronic pain is a condition that is produced by the brain. So the brain produces this massive emotional impression an aversive kind of feeling that pain is associated with, but this is something the brain makes itself. And so that's why we wanted to look at this region, because this region is involved in emotional aspects, prediction, avoidance of pain stimuli, motor response. And there's a lot of studies that show that there are changes going on in this area under neuropathic pain in specific. And one particularly interesting observation that has been made in humans, but also in rat models, is that the cortical thickness here in this area changes in patients with chronic pain. So this is the anterior and the mid-cingulate cortex, and you can see these are the two only specific regions. Also often people find these effects in the insula, but in these areas you can observe a decreased cortical thickness, whatever that means. It's volume, voxel-based morphometry using MRT approaches. Nobody knows exactly what that means, but there is this kind of change in density, and this is actually reversible. So if patients get out of the chronic pain state, that goes back to normal. That could be reproduced here in rats, where like a chronic pain paradigm, neuropathic pain model was applied, and one can see the same changes here happening like in humans. And this was another sort of big reason for us to ask whether in this area there might be changes in the dendritic tree composition, spine density, and so on, and how those changes, those plastic changes, would correlate, for example, with the dynamic development of the pain phenotype, which cells are responsible or showing these kind of effects. And of course, what in the end of things, we really would want to make the point that these structural changes are actually causal to producing the pain phenotype. So this is where we want to get at, but this we haven't yet achieved. So in order to address this, we're using a longitudinal correlative approach, which is depicted here. So the mouse sort of is first imaged in baseline using these chronic windows that I showed before and two-photon microscopy. So we can assess pain behavior, withdrawal reflexes, for example, and we establish a baseline here. And then we can add this noxious signal, for example, spared nerve injury, and then we keep imaging behavior over a long time period, over months. So we have done this up to 9 months after the noxious stimulus. And this, of course, is a correlative approach. So if we see the development of the phenotype, and we do see at the same time changes in structural plasticity of the dendritic tree of these neurons, then of course one could say, well, those two things are correlated, but we still don't know whether it's causal. So the way we do this is we again use viral gene transfer into this area here of the cingulate cortex. And in some cases, actually, in the meantime, in all cases, we also inject the thalamus down here to have also presynaptic inputs. And we're actually looking not only at the plasticity of the dendritic tree, but we also study plasticity of presynaptic inputs, so we can do both at the same time. But I'm not going to go further into this. But this is what you saw very initially when I explained two-photon microscopy. Remember the stack that was going into the brain? This was the situation with both pre- and postsynaptic elements labeled. So our goal here is not to do like some kind of stereological or statistical analysis in terms of looking at small dendritic segments, but our goal is to study entire neurons. So again, we have the sparse labeling technique where we have only individual neurons sitting in a certain imaging volume, and we image the entire full neuron, as you will see. And then, of course, analysis is really the key in this project. I mean, we spent now 2.5 years of developing an automated workflow, and I think it's now actually working. So we can now put in a cell and we get sort of the tracing result out. So you have to basically first crop these stacks, then align them at different time points, do this 3D volume registration, then segment the data, and then compare basically the different time points in that imaging experiment. So what we're using here in terms of chronic pain model is chronic neuropathic pain, spared nerve injury, which is depicted here. So one cuts basically 2 parts here of the the saphenous, the sural and the tibial nerve, and the saphenous nerve stays intact, and that leads to a very strong and chronic pain development, chronic pain development. And as a control, we use sham operations where we don't do this cut. And as behavioral analysis, we use here this kind of withdrawal responses with von Frey filaments. So you poke the skin and you look for the withdrawal of the paw, and you can use different strengths of these filaments to then get a quantitative assessment. So this is an example where we have now 3 neurons in our imaging volume. They are still reasonably separate from each other so that they can be analyzed. You can also see that they actually differ in their morphology quite a bit. So this one is much more spiny than the green one, for example. And we can, of course, try to do morphological categorization of these neurons and relate the overall morphology to changes in spine plasticity. And of course, we also do post hoc analyses. So we find these neurons again after sacrificing the mice and do immunohistochemistry so that we can identify what kind of cell types these are. So this is the data that needs to be analyzed. And here is just an example of spine changes after SNI. So this is a basal state here, then 2 weeks and 10-week state. And you can see there are some spines that are quite stable, yet after 10 weeks they're gone. This group of spines here is kind of quite stable. But overall, if you compare this stretch with that one, there's a pretty strong loss of spines over this time period. Now, as I said, we're looking at full neurons here. Now, what we can do is we can compare the basal state with, for example, the day 3 state, so 3 days after the spinal nerve injury, and then simply color the areas of spine density changes in this color code. So basically, dark bluish colors would mean that there's a decrease in spine density, and bright colors would mean there's an increase in spine density. And what this analysis actually shows, I think also for the first time, is that these kind of spine density changes are actually very much organized. So it's not that this is kind of randomly occurring across the whole neuron, but there are zones of plastic areas like these ones here, for example, that show like an upregulation of spine density. It's kind of small segments of typically 10, 15, 20 microns length, and most of that neuron actually doesn't change. So this would be these colors here would indicate no changes in spine density, and these kind of bluer colors here that could be would be basically decreased spine density. So you can see this kind of plasticity, like the spine density plasticity, is actually organized typically in segments. And these segments, of course, could correspond to specific inputs that these segments get from other areas of the brain that then might be more plastic than others and might be more related to the pain phenotype than others. So this shows now the analysis over 4 months. So the same neuron imaged over and over again over a time period of half a year. And the code here, the color code is always related to the basal state. And what we can derive from this is that at 3 days, we have a lot of areas with upregulation of spine densities. But then if you go along that time development, we can see that after 4 months, we actually observe spine loss as the dominant effect here. And I think this is very nicely correlating with the voxel-based morphometry results that people have in humans and in rats. We are doing this actually in mice right now too, where one sees that after 2 months, so basically around this time period here, the voxel-based morphometry gets kind of significant. You can see the volume changes in the cortex. So that correlates actually very well with this kind of late phase of spine decrease, overall spine decrease, which is sort of preceded by an early response with increased spine densities. And so this is just from the same animals, the behavior, and we can see after 3 days, the hypersensitivity to von Frey responses is maximal, as you can see here. So this data has shown that structural plasticity in neurons of the cingulate cortex correlates with chronic neuropathic pain states. And we have also shown that such plasticity changes of spine density actually are highly organized across the entire neuron. So maybe these segments could be something like processing units of the dendrite, or it could be sort of pain-specific inputs that impinge onto these segments. So that was the pain story, and to come back to my global theme here, so conclusions regarding cell biology at work. So I think in vivo studies are clearly feasible, and they are essential to extrapolate from in vitro to in vivo. I think in the dense core vesicle, we have seen that things match up quite well, but that there are still additional things to be learned. Yeah, so you can typically add aspects that you cannot identify in less complex model systems, but one has to also then conclude that strongly conserved mechanisms such as organelle transport, which is basically conserved from Drosophila to human, such mechanisms, I guess, one can actually extrapolate very reliably. Last not least, the in vivo studies provide a possibility, of course, to use physiological stimuli to assess basically their impact on biological mechanisms. And that, of course, in reduced systems is always a bit difficult to use physiologically proper stimuli. So this work was done in collaboration with different groups. So the dense core vesicle story was done mostly by Johannes Knabbe and Joris Nassau. So Johannes is now a resident in psychiatry, so he was an MD working in the lab, and he established all the dense core vesicle imaging together with Joris, a master student. And this dense core vesicle project was a collaboration with Matthijs Verhagen in Amsterdam, and Joris is now moved to Amsterdam because he discovered his love for dense core vesicles. Juan Boffi, an extremely talented Argentinian postdoc, did all the work on the chloride imaging that I showed you, and the pain work was done on the analysis side by Hong Wei, who developed all these automated workflows, and Sanjeev Kaushalia did the imaging of the mice, and this was a close collaboration with the department of my wife, and I thank you for listening.
Thank you very much, Thomas, for this amazing talk. I think we have time for 1-2 questions.
Thank you very much. A very nice talk. So I'm coming back to the very beginning. You actually said in the beginning that dense core vesicles can only be synthesized at the cell soma. So what about like Golgi outposts or and then actually also showed evidence of fusion of dense core vesicles. So is that fusion happening, and is that somehow regulated? And is there actually, is there a sites more distant from the soma where you could actually generate large dense core vesicles?
Yeah, I mean, I'm also convinced that there are Golgi outposts. This is actually also something we're studying with one PhD student working on this to really do correlative EM and fluorescence microscopy to really nail it down. I think they exist, but it seems that they're only in a very close sort of 100 microns away from the soma. So further out, at least we have not seen them. So sure, I mean, there one could expect that you can get budding and maybe even recycling of dense cores. But to my knowledge, this has not been really looked at. I mean, even the Golgi outposts are very poorly studied. There's not many people working on this, to my knowledge at least. I think it's very interesting to address this, but yeah, I think it's a good possibility that this really happens. Now, in terms of the factors that are required to attract dense core vesicles, this is something that Matthijs is, of course, studying a lot, and I mean, they have identified clearly docking molecules that are required for dense core vesicle docking and fusion. So I think there will be more on that shortly. So what the underlying molecular underpinnings are that label the sites of DCV release.
Thank you for the fascinating talk. Did you happen to maybe test whether the speed of DCVs in the axons pick up when there is synaptic plasticity, for example?
That's a very good question. So, yeah, we haven't looked at that yet because we would have to generate a situation where we know that now plasticity is going on, and we would have to then specifically record DCVs in the same area, maybe the same axon that is involved in plasticity. Yeah, I think that's hard to achieve, but maybe not impossible. But that's, I mean, I think that's definitely maybe one could, like chronic pain. I mean, this to induce like a neuropathic pain model. I mean, that obviously is a very, very strong stimulus for plasticity. So one could do it like this, for example, introduce the neuropathic pain model and then immediately image and see what happens to the dense core vesicles. Is there more fusion? Is there more extensive transport? Is it more into one direction and so on? I think that would be definitely feasible. That's a good point.
Thank you
hello.
very much.
Thank you. Thank you for a very interesting talk. I have a question concerning the dense core vesicles, because you said that you've tested them under awake and anesthesia. And my question is, what type of anesthetics did you use? Have you tried like different ones, and did you receive similar results?
No, we only used isoflurane for this. We didn't use other anesthetics.
Okay, because you said that there were no difference in the mean of speed and in the maximum of speed, but there were big differences in directionality.
Right.
Yeah. Do you have any idea why?
Unfortunately not.
Okay.
I have no idea what could be the basis for that.
Yeah, because I think that it is interesting whether the same results will be with different anesthetics, for example, or whether it's only isoflurane doing something, or like in general anesthesia is shutting down some pathways, and that's why.
Yeah. I mean, also I think what we were discussing, what you mentioned that BDNF reacts very quickly to isoflurane anesthesia, so that of course could be relinked. I mean, maybe there's as you ramp up the isoflurane, maybe there's a very acute phase of increased release. But I mean, so we did several of these experiments where you basically first have anesthetized and then you wake them up, and then one should see in that phase the difference. But as far as I remember, they looked at this, but they couldn't see any effects on the trafficking in this period. And you can do, of course, the reverse. I mean, start with the awake and then kick in the anesthesia. But I think also there, as far as I remember, they looked at this, but I don't think there was anything.
Okay, thank you very much.
Okay, we will stop here. Thank you very much, Thomas. And now we have a few minute break, and we're coming back at 11 for the next session on cortical plasticity.